exam for TJ, and I'll just let you take it away from here. About how long is your presentation, would you say?
Yeah, about 35 to 45 minutes.
35, 45. Okay, great. Sounds good.
Yes, I did start the recording. I'm not sure where that's going to be recording to, so hopefully that goes to your cloud, Vince, and we can disperse that as needed.
Yeah, I think it should, because this is my Zoom room, so that should be what we want.
Great.
So, okay,
Great.
So I can give you the link when I get it later. So
awesome. Then I will go ahead and jump into it. Thank you for everybody for attending my proposal talk. I'd like to start out by thanking Dr. Vince Calhoun, Dr. Sergey Pliss, Dr. Matthew Block for serving on my thesis committee, and Shella, Kyle Holtz, and Jeffrey Davis for serving as my advisors over the last several years. My name is Theodore LaGrow, and the title of my PhD thesis proposal is Exploration of Spatiotemporal Dynamics in Neurodegenerative Functional Brain Networks. This afternoon, I will cover the background of my thesis research, my dedicated aims, the timeline of the remaining research, and some acknowledgements. Resting state functional magnetic resonance imaging, or resting state fMRI, consists of scanning the brain when subjects are at rest but not asleep. Within fundamental science and clinical settings, resting state fMRI has revolutionized our understanding of the brain function and organization. The technique relies on the BOLD signal, which stands for the blood oxygen level dependent signal, and is an indirect measure of neural activity that relies on magnetic properties of the oxygenated blood in the brain. Throughout the principle of neurovascular coupling, we are able to use this measurement to record large-scale brain activity over time. This approach has since become a valuable tool for investigating functional connectivity within and between brain networks, and has led to important discoveries about the brain's intrinsic organization and its alterations in various neurological and psychiatric disorders. Resting state fMRI studies have shown that brain activity is not just localized, but there exists large-scale coordinated activity across the brain. One example of this is resting state networks, which are networks of the brain structure that have been consistently shown to exhibit similar activity at rest. To go further, when the data is organized in canonical resting state brain networks, shown here at the top displaying scans from a cohort of concatenated healthy subjects, we can see empirical examples of repeated patterns of widespread coordination in the brain over time, as shown by the semi-repeated pulsations. Resting state connectivity is typically obtained from this data by comparing the synchronized fluctuations of the BOLD signal between different brain regions, often achieved by parcellating the brain into regions of interest, or ROIs, using an atlas or seed-based approach, which provides an effective measure of the strength of functional connections. The BOLD Central is central to this research, and it provides a non-invasive clinical measure of brain function and connection. The lack of task makes resting state functional for fMRI particularly attractive for patients who have difficulty with task instructions for evaluating cognitive decline. Resting state fMRI is a non-invasive and relatively less expensive modality than other brain imaging techniques, allowing for more frequent scans for patients with higher disease propensities on a longitudinal scale. Thus, the application of resting state fMRI in research and clinical settings have been growing for the past 2 decades. Alzheimer's disease is a neurodegenerative disorder characterized by severe cognitive decline and a decrease in neural connectivity in brain tissue. While many therapeutic approaches exist today to help slow Alzheimer's progression, medical communities continue to struggle with adequate prevention and treatment of strategies. As it stands, Alzheimer's disease remains one of the largest threats to the public health, impacting over 50 million people worldwide, as reported by the World Health Organization this year. Alzheimer's disease is by far the most common type of dementia, estimated to account for 60 to 70% of all dementia cases. An estimated 6.5 million Americans ages 65 or older are living with Alzheimer's dementia today, with the number projected to grow to 13.8 million by 2060. The incidence of progression to later stages of Alzheimer's disease increases year over year. Moreover, the costs related to Alzheimer's disease are enormous, estimated to reach $345 billion in 2023 in the US alone. The demographic composition of this population is skewed towards an increasingly proportion of the elderly, and related costs are predicted to more than triple by the year 2050. Without medical breakthrough to prevent, slow, or cure Alzheimer's disease, early detection is more important than ever. If dementia is discovered early in the disease development, the number of Alzheimer's disease incidents can dampen by nearly one third through rehabilitation exercises and medication. By identifying Alzheimer's disease early, we can ensure that the growing population of elderly patients receive the care they need and provide fundamental knowledge to help slow and eventually prevent the disease progression. Most investigators believe that Alzheimer's is a slowly evolving process, likely beginning years to decades before the clinical symptoms manifest. Due to the severity of effects caused by the disease, there is a strong interest in identifying individuals at an early stage. To clinically discern the progression of dementia, cognitive performance exams are administered in conjunction with subject-specific demographic information. Mild cognitive impairment, or MCI, represents an attempt to characterize subjects at an early clinical phase and has been the target for many clinical trials. In recent years, while analyzing the progression of dementia of Alzheimer's type, or DAT, several researchers have assessed subjects in specific subgroups based on longitudinal progression. These groups will be the focus of the present research. The first three groups, SNC, UNC, and SMCI, represent subjects without dementia of Alzheimer's type related pathophysiological changes. These subjects do not have a diagnosis of Alzheimer's during the study window. The remaining three subgroups, PMCI, EDAT, and SDAT, represent the dementia of Alzheimer's type spectrum that spans from clinically asymptomatic to severely impaired. When thinking of how to pre-process this data, pre-processing of resting state fMRI involves a series of steps to clean and normalize data to prepare it for subsequent analysis. The general pre-processing pipeline involves the following steps, many of which are study-dependent, where this list is not exhaustive of everything that could be done, but generally considered to be the bare minimum. Temporal filtering, physiological noise removal, motion and other artifact corrections, and the choice of atlas registration remain controversial and contentious topics, as many times siloed in research groups where specific parameters are previously cited through lines of research, leaving open the question of robustness and fidelity of specific network interactions and disease implications. One of the more controversial pre-processing steps is the globally averaged signal, or the global signal, where it is often used as a way to decrease noise in a data set. The most common way to do this is the global signal regression. The rationale behind using GSR is that neural activity is localized, and thus any signal that is widely coherent is likely to come from non-neural processes. Although GSR has shown to decrease various types of noises and artifacts, other research demonstrates widespread resting state networks, much like resting state signals, much like the resting state networks, and say that global signal regression could be eliminating important neural information. New work continues to progress our understanding. However, this remains an open question that I hope to help contribute. To better understand how these resting state networks might interact, how these networks, and especially how they interact over time, spatiotemporal patterns are used for dynamic network analysis. These semi-consistent or quasi-periodic waves of activity, known as quasi-periodic patterns, or for short QPPs, were initially discovered by our lab in rats and have since been discovered in mice, humans, and non-human primates. Here demonstrated is the algorithm illustration of the QPP approach, followed by demonstrations of signals in humans and rodents. This pattern is most notably characterized by the anti-correlated signal between the default mode network and the task-positive network. QPPs contribute to a general whole brain functional connectivity of the brain, which can be altered by various diseases. The objective of my PhD thesis is to harness the capabilities of spatiotemporal analysis methods to understand the progression of Alzheimer's disease using functional brain dynamics. I'm planning to do this by achieving two aims. First, I plan to identify biomarker differences of ultra-slow, widespread spatiotemporal brain patterns in Alzheimer's disease progression. I'm proposing to carry this first aim out through the fulfillment of two sub-aims, looking at the fidelity of quasi-periodic patterns and the effects of Alzheimer's disease progression on these patterns. Secondly, I plan to quantify disparities in Alzheimer's disease progression via global signal phase coupled variations. In this first aim, I plan to identify biomarker differences of ultra-slow, widespread spatiotemporal brain patterns in Alzheimer's disease progression. The goal of this aim is to explore specific frequencies of QPP signal, further generalize QPPs in a diverse data analysis, and then identify differences in QPPs in neurodegenerative disease groups. I hypothesize that the combining signals of slow 5 and slow 4 frequencies will play a dominant role in the detection of the delineation of the characteristics of this signal within the resting state fMRI bold signal. There will also be a disruption of the anti-correlated signal through disease progression. I will carry out this analysis that identifies differences pertaining to the spatiotemporal brain patterns during the progression of AD. To the left are the objectives that I hope to complete as we go through the course of this aim. In this sub-aim, I plan to further generalize the use of spatiotemporal patterns in dynamic brain state analysis with the fidelity of quasi-periodic patterns. QPPs are reliably recurring spatiotemporal patterns of propagating activity. QPPs contribute to normal functional connectivity, typically measured by the level of anticorrelation between the default mode network and the task-positive network. The contribution of QPPs to typical functional connectivity in attentional networks may shed light onto the etiology of disease and brain states related to disrupted attention. Here, I am displaying the current analysis that can be performed utilizing this spatiotemporal signal of the QPP. These analyses allow for the understanding and potential use of biomarkers for subjects with neurodegenerative underpinnings. For example, the data is pulled from this data has been pulled from a publicly available open source dataset known as ADNI. Displayed here are 4 subjects with a total of 9 longitudinal scans. For reference, here is displayed to the top template for each quasi-periodic pattern, the reverse phase of each of the 3 quasi-periodic patterns, the spectral density plot, frequencies of the QPP occurrence, signals before QPP regression and after QPP regression, signal correlation histograms for each of the signals, functional connectivity of the brain before and after QPP regression, waveform plots of the subnetwork correlation, global signal, and subcortical global signal and QPP correlation. Additionally, we can look at the same outputs without the use of global signal regression to compare the various findings. As previously reported by our lab, the spatiotemporal signal accounts for a large portion of the functional connectivity when regressed from the overall functional connectivity. Further, we can look at the global signal, subcortical global signal, and non-global signal regress component of QPP1 to further find the coherence of each of these signals. As an example, this result does show that the QPP1 does correlate strongly with the global signal, giving coherence to these findings. However, to better understand the generalized use of QPPs, several questions arise. The selection of pre-processing procedures, including imaging parameters, the choice of bandpass filter, voxel to network or region of interest atlas delineation, and other relevant hand-tuned or empirical factors play a pivotal role in shaping the trajectory of research within the field of resting state fMRI. In particular, these choices often become a defining feature when referring to prior studies conducted within the same laboratory or research group, where researchers frequently employ phrases such as used in previous studies to establish continuity and build upon existing knowledge. These questions pertain to TR, or the repetition time of an fMRI scan, denoting the time it takes to image a single volume of the brain, the atlas of choice to register the brain scans, and the frequency range linked to the bold signal neurovasculature coupling. To analyze the TR's role in intrinsic brain dynamics, this objective explores the influence of the repetition time on dynamics of QPPs. With longer TRs, there is higher chance of aliasing the anti-correlated signal. However, the anti-correlation between default mode network and the task-positive network will be preserved at faster TRs. We will be using the Human Connectome project as displayed here, where a large corpus of data of relatively faster scans of TR of 0.72 seconds will be utilized. Displayed are the demographics of the dataset curated by 2 pipelines, both using NeuroMark and CPAP. For current analysis, I've delineated the groups into 5 for variations of volume removal to simulate longer TRs. The known ideal length for a QPP detection is 24 seconds, which then deprecates the number of volumes needed to detect the signal in a reduced number of window lengths or the number of brain volumes to be detected by this signal. From these findings, from initial findings, there is consistency between resampling of TRs, and slow 5 and slow 4 frequency bands do seem to retain the strongest anti-correlation between higher TRs. To demonstrate some results, here are the 5 groups of resampled Human Connectome project data for detection of QPPs. Of note, the QPP anti-correlation between the default node network and the task-positive network is preserved. even when deprecated TRs of 3.6 seconds. Moving on, taking the atlas perspective, this objective focuses on how brain atlas choice affects QPP detection in functional brain connectivity analysis. We first scrutinize the fidelity and consistency of the proposed method across various canonical brain atlases used in resting state fMRI network studies, encompassing atlases based on anatomical landmarks, gyri sulci structures, functional connectivity, functional mode dictionaries, and adaptive subject-specific atlases. Many of these atlases are comparatively evaluated for the first time. Further, we delve into spatial remapping of QPP signals onto a common template, specifically the NNI 152 template, investigating spatial consistency across parcellations and examining potential discrepancies due to anatomical references choice. Although not shown here for brevity, some comprehensive analysis underlines the pivotal role in brain atlas and functional connectivity characterization, emphasizing the importance, especially when comparing results across different data sets. The aim is to shed light on the atlas dependency of QPPs, pinpointing the consistency of variations across different parcellations. Here, I provide an example illustration of each of the network parcellation used in this study for the 11 human brain atlases of interest. Although this is not an exhaustive list of all potential atlases, each are widely used in their respective lines of research and hold potential to evaluate various characteristics of resting state fMRI analysis. Looking at the frequency's significance, the third objective aims to uncover the role of frequency bands in QPP detection and their subsequent influence on functional connectivity analysis. This exploration seeks to address the pivotal question: how does the selection of frequency bands impact the identification and interpretation of QPPs? Using the full signal, this is the full bandpass interest of neurologically and physiological reference of the signal. The infraflow signal is the most commonly used frequency range for resting state fMRI analysis, reflecting the spontaneous neural activity and functional connectivity in the brain. It has been further divided into multiple distinct frequency bands based off of neurophysiological studies. Slope 4 and 5 are considered to be more specific to neural activity and functional connectivity, whereas slope 3 and 2 are less explored signals due to the low sampling frequency of fMRI, but have been suggested to be related to neural oscillations, potential cognitive processes. May also be influenced by cardiac and respiratory signals. As an initial show, the initial QPP bandpass filter analysis demonstrates 8 groups of various bandpass filters. All bandpass filters exhibit a strong anti-correlation between the default mode network and the dorsal attention network, one of the subnetworks of the task positive, with slope 5 representing the strongest anti-correlation. Slope 3 trunk, slope 3, slope 2 do not demonstrate correct phasing of the signal, as the cycle times seemingly resemble more cardiac pulsations and respiratory cycles. All 8 bandpass filters without global signal regression applied on the bottom column have a correlation between QPP1 and the global signal, which is nearly identical over all bandpass filter choices. The preservation of QPPs amidst various pre-processing considerations emphasizes their robustness and highlight the potential as a critical marker of intrinsic brain dynamics and functional connectivity. Thus, while the field of resting state fMRI embraces a range of pre-processing options, QPPs serve as an underlying element that warrants further investigation and exploration in future research endeavors. Some pitfalls of this aim would be if I am unable to identify QPP patterns in well-known and consistently used brain atlases. However, this will still prove interesting, as this will be the first comparison of many atlases to use for specific design choices and suggestions. The outcomes of this research aim will be a journal manuscript either to Imaging Neuroscience or Frontiers in Neuroscience. Moving on, in this aim, I plan to analyze the effects of Alzheimer's disease progression on spatiotemporal resting state fMRI brain patterns. Alzheimer's disease poses a significant challenge due to its detrimental impact and the difficulty of early detection, often resulting in irreversible progression and severe side effects. One of the primary obstacles in diagnosing Alzheimer's lies in the limited availability of non-invasive methods for assessing the disease dynamic and multifaceted nature. Research conducted by Anzur Abbas in our lab demonstrated a diminished role of QPPs in functional connectivity of individuals diagnosed with ADHD. a similar but very different neurodegenerative disorder. Similarly, Michael Beloy also investigated in mice, where he revealed a decreased involvement of QPPs in functional connectivity of rodents affected by Alzheimer's disease. Strong functional connectivity changes occurred both in neurodegenerative disorders, as well as the intrinsic contribution of QPPs to this connectivity, suggesting that employing this method offers more comprehensive understanding of the underlying mechanisms and potentially serve as a more effective clinical biomarker. Results from this study warrant clinical explanation of neurodegenerative diseases. In this study, we will leverage longitudinal Alzheimer's data to investigate the differences among discrete milestones of Alzheimer's progression. By analyzing large open source data sets, we aim to unveil fundamental and robust characteristic differences that can distinguish individuals at different stages of the disease. The utilization of longitudinal data enables the exploration of temporal patterns and spatiotemporal dynamics, shedding light on the evolving nature of Alzheimer's over time. The investigation of discrete stages of Alzheimer's progression also offers several advantages. Firstly, it allows for the identification of critical transition points and inflection moments where the disease trajectory may change significantly. Understanding these turning points is critical for developing interventions that can effectively delay or mitigate the disease progression. Furthermore, the analysis of the distinct stages enables the identification of specific biomarkers, imaging patterns, or clinical indicators that are particularly relevant to each stage. This information can greatly enhance early detection, monitoring efforts, and improve patient outcomes by facilitating timely interventions. To analyze distinct milestones of the disease progression, here displayed are the demographics of the three-way classification for Alzheimer's, curated by the Neuromark Initiative. This data provides rich corpus of previously utilized data to help compare across stages of Alzheimer's disease. Further, I've curated data myself from the ADNI database to demonstrate initial findings. To highlight the value of this data, I have relatively equal scans from both male and female participants and a larger corpus of overall scans, 86 scans to be exact. This data has helped demonstrate findings suggesting a disparity of dynamic functional connectivity across the stable progression of Alzheimer's disease, warranting further investigation. For completeness, this is the overall data set curated for stable diagnosis of Alzheimer's patients in the ADNI longitudinal database. Due to the complexity of the data, the required time, and careful planning, many studies do stray away from looking at this longitudinal data due to the nature of the disease with the complexity of the data set. The data set will allow for the characterization of widespread changes from different stages of Alzheimer's disease progression to be analyzed. Within the second research group of the longitudinal Alzheimer's data, there is rich information to glean. Here I demonstrate a histogram of the available scans from the longitudinal subjects who changed diagnosis from either normal cognitive to mild cognitive impaired, or from mild cognitive impaired to dementia of Alzheimer's type during the ADNI research project window. The red line here indicates month 0, or the flexion point in diagnosis, and each bin in the histogram is 12 months, representing an unexplored pathology of clinical milestones in disease progression. Here I have demonstration of group differences with waveform plots. Each QPP template is groupwise independent with its each own template. The templates for the normal cognitive and the dementia of Alzheimer's types demonstrates an initial degradation of the anti-correlated signal between groups, with the slow 5-pass band filter exhibiting the best demonstration of anti-correlation differences between the networks over groups. Slow 5 and slow 4 illustrated delineation of the infraslow band fast band filter, offering potential further insight into network interaction over the disease. In summary, for this aim, I am striving to identify the effects of Alzheimer's disease progression on spatiotemporal resting state fMRI brain patterns. Through this, I have been able to process the data, organize it into groups, utilize some spatiotemporal patterns, and use this on a subset of the ADNI data. Currently, I am processing and finishing up the larger data set and comparing this on stable groups. There are some potential pitfalls where we are unable to identify significant differences between these patterns in the disease progression. However, this will still be useful information as the longitudinal data is often overlooked due to data difficulties, and the resulting analysis will be well received by the larger community. The outcomes for this part of the research will be a journal manuscript either to Imaging Neuroscience or Frontiers in Neuroscience. Moving forward, in the second aim, I plan to quantify disparities in Alzheimer's disease via the global signal phase-coupled variation. The goal of this aim is to truly identify different differences in Alzheimer's disease progression utilizing the global signal that may be phase-coupled with brain states within resting state fMRI scans. I've hypothesized that the progression of Alzheimer's may be disrupted within the relationship of the global signal and co-activation patterns. Alzheimer's pathology might be impacted by dynamic brain region interactions. Here, I have also listed a complete list of tasks that I will need to complete through this. I will carry out the analysis that identifies differences pertaining to the globally phase-coupled variation in brain states under healthy and neurodegenerative subjects. Resting state functional magnetic resonance imaging, RSFMRI, is a powerful tool for investigating these intrinsic functional networks of the human brain. However, conventional analysis of resting state fMRI data usually assumes that functional connectivity between brain regions is static over time, neglecting the dynamic aspects of brain activity. Co-activation patterns, or CAPS, are a method for extracting transient brain states from resting state data at a single time frame resolution. CAPS can reveal the temporally variability in the complexity of the brain interactions that are not captured by static functional connectivity analysis. CAPS can also provide insights to neural origins and behavioral relevance of resting state fMRI signal. CAPS and fMRI analysis offer a range of benefits and hold significant importance in the study of brain function connectivity. These patterns serve as a valuable tool for identifying and characterizing distinct brain networks and their dynamic interactions. For instance, well-known networks like the default mode network, task-positive network, saliency network, and dorsal attention network can be effectively delineated and studied through CAPS. Furthermore, the relationship between brain states and cognitive functions, encompassing intention, memory, emotions, and consciousness, can be explored by leveraging CAPS. Additionally, CAPS facilitate the detection, diagnosis, and understanding of various brain disorders, including Alzheimer's disease detection. The predictive potential of CAPS extends to behavioral outcomes and individual differences, aiding in the projection of cognitive performance, personal traits, and clinical symptoms. However, the use of co-activation patterns in fMRI analysis offers benefits, but also faces methodological challenges. The choice of seed region, thresholds, clustering methods, and the cluster number can influence CAP outcomes. CAPS might not always indicate causal brain region interactions, limiting the understanding of information flow. Factors such as physiological noise, head motion, and global signal fluctuations can affect CAPS reliability. Additionally, CAPS consistency can differ across datasets, equipment, protocols, and analysis design, necessitating careful interpretation of this analysis. One possible way to evaluate CAPS is to examine their relationship with the global signal, which represents the average of all voxels in the brain. The global signal is often considered to be a nuanced factor that reflects physiological noise or scanner effects that is usually removed from fMRI data before analysis. Contrarily, recent studies have suggested that global signal may also contain meaningful information about large-scale brain dynamics and cognitive performance. Here demonstrated are the first several QPPs templates from the data that is global signal retained and removed. The global signal can be broken down into distinct signal phases and correspond to different brain activities, potentially reflecting various aspects of brain function. In the supplementary section of a 2022 Nature paper by a previous postdoc in our lab, the authors conducted a comparison involving complex principal component analysis, or PCA, a technique similar to QPPs. Notably, this correlation between their pattern 1 and the global signal appears almost indistinguishable. We are able to replicate these findings using QPPs, observing a similar correlation between QPP1 and the global signal. This points to a specific link between the global signal and its potential influence on functional brain activity. As a result, it becomes intriguing to explore how CAPs are influenced by the phase and frequency of global signal, warranting further explanation into implications of neurodegenerative biomarkers. Here demonstrated is an initial overview of current replication findings in the understanding of functional brain states in relationship to the global signal phase. This research centers around a study conducted by Gutierrez in 2019, where the authors explored the dynamic patterns of brain activity in rats, utilizing phase encoding, as they say, of functional brain states. Here demonstrated are the results from the paper, which reveals distinct co-activation patterns occurring at specific phases of the global signal across the circular dispersion plots. For clarification here, we denote the terminology of the relationship as phase coupled rather than phase encoded to avoid confusion with classical MR terminology. Our work extends and validates these findings using human resting state fMRI data. To further enrich our investigation, we explored variations in the number of brain states using resting state fMRI data, where here depicted are the results of our analysis. Fully replicating Gutierrez and al.'s approach, we first examined brain states with 6 components, or K equals 6. By visual inspection, we can see distinct separation of global signal phases. This replication suggests a potential of small number of brain state clusters encoded by the global signal. Next, we simplify this analysis by considering 2 distinct brain states, K equals 2. Zooming in here, the separation of these states is highlighted by the clear distinction in the absolute phase values of the global signal. Here, the blot plots are similarly representative of the circular dispersion plots of the phase-coupled brain states to the global signal. These plots demonstrate a clear separability of the distinct brain states encoded by the global signal using the most common or simplest case of K equals 2. Finally, with 3 brain states, or K equals 3, we employed additional analysis using an ANOVA test to confirm the separation of functional brain states based on the phase of the global signal. We do not see a separation of groups of the green and maroon dots. However, here there is a separation when adding into the blue points, indicating a separation of brain states in a low number via the phase coupling of the global signal. Due to the use of specific data being the ADNI data and a slower TR, this study warrants further investigation into the relationship of global signal and the intrinsic brain states with various data sets. In summary, our work not only replicates and extends the findings of Gutierrez, but it is also introduced to new insights to the relationship between functional brain states and the global signal phase using human resting state fMRI data. These results emphasize the importance of considering the dynamic interplay between brain states and the global signal phase, offering a promising avenue for understanding the complexity of brain function and its potential application in various neurological contexts. Through this work, I will produce a review paper on the global signal regression and resting state fMRI, discussing the state of the art, key debates, future directions, and its connection to the bold signals neural correlate. The review paper will offer updates and insights into the guidelines for global signal regression use. Currently, this paper is in preparation. I will also produce a conference paper to highlight initial discussions on the results, aiming at OHBM 2024. And further, this work will be encompassed into an IEEE conference paper, where the work will be disseminated to a larger community of computational neuroscientists. In conclusion, each of these aims highlight the tasks needed to complete my thesis aims. Here, highlighted in yellow, are the tasks which are currently underway and very close to being done. Green is indicating the full extent of processed and complete tasks, where the dark black does indicate tasks remain to be done. Both of my aims contribute to the exploration of spatiotemporal dynamics within neurodegenerative disease progression. Much of this work is far along and will not take long to disseminate to the greater research community. In terms of contributions to the field, I have summarized the projected first author contributions of my thesis work. By the time of my thesis defense talk, I hope to have contributed 2 journal papers, 1 review paper, 1 conference poster, and a conference paper. Although aggressive, I do plan to complete this work within the next 6 months to finish my thesis work. Much of the preparatory work and data curation is complete, which encompasses a major time hurdle. Each of these aims are laid out in terms of the major milestones that need to be completed. To summarize my previous grad school work, I have made major contributions to review papers, original research, and grant work. Although related in topic, many of these research lines are supplementary or utilized by analysis required by my thesis work. Previous work does involve cross-species fMRI comparison, global signal characterization, software dissemination, bold signal characterization, and large data automatic cell detection. Further, I contributed to several conference submissions, both relevant to the current research line and previous grad school research lines. Finally, I earned a full patent for my work in developing the technology for a company founded during grad school. This work is not done in isolation. There really is too many people to acknowledge and thank for helping me over the last several years. I'd be remiss if I didn't mention the Cal Holtz Mind Lab and everybody involved. Each of you have contributed much to this research and generally helped my mental state day to day. People at the Trend Center are great collaborators, and both Eloy and Vaibhavi are great friends. The Graduate Teaching Fellows, Grand Challenges, and the George F. Riley Fellowship, I greatly thank for funding and help in my education throughout this time. Lastly, I would very much like to thank my family, my mom, dad, my sister Virginia, my fiance Jennifer, and both my aunt and uncle and my grandfather for constantly being there and inspiring me to continue on through the PhD work. And with that, I will open the floor to questions.
Great, thank you. I think we have Sergey, you joined, yeah? Welcome.
Yes, I'm here. I apologize for being late. I started at aim 1, so I got the gist.
Yeah, great. While I've got you then, did maybe we can just start with you if you have any questions for TJ?
TJ, can you, I'm a little confused by what a CAP is and how it relates to FNC, functional network connectivity. Your first slide especially where you introduced it, where the spatial maps are changing. You're saying you use seed correlation to compute it, and at the same time you are saying that they are instantaneous. So what kind of information enters to compute the correlation between voxels? Yes, that is the slide. Thank you.
Yeah, so this is a demonstration of the potential use of CAPs. We can use a seed-based approach, specifically when looking at specific networks like the default mode network. We can initialize in the PCC a well-known region of the default mode network to find the connectivity between various regions. We can also do a group-wide correlation to find the CAPs at all seed level, using each one of the brain states to better encapsulate the co-activation patterns between the brain states. So finding the instantaneous change of phase within the global signal has at least shown various differences between diseases. What I don't show here is that previous work in rats has shown that CAPs do have differences between disease states while using this method.
Well, I think I'm getting really technical trying to understand how you compute it, but if you take one voxel and correlate the time series of that voxel with every other voxel, then you are using the entire fMRI time series. Are you windowing this time series to compute the correlation somehow? How do you get the dynamics out of this?
Yes, so it is still single time point evaluation using these CAPs. If I'm understanding correctly, to find the phase of each of these, we can take the Hilbert transform at each of the distinct time points to get the phase of the signal or each of the brain volumes at that given time point.
Okay, so this is kind of a phase coherence maps then, right? Not correlation, even though you have this speed.
Yes. Yes. Perhaps the nomenclature is incorrect here, but yeah, maybe we can talk about that.
There is no correct or incorrect. It's just I'm trying to map it to my language, my internal model of what's going on. Okay, thank you.
Are you selecting peak time points to do this, or are you? Is that where you get? Because you mentioned individual time points.
Yes, so that has been a high point of contention, especially I know with your group demonstrating that connectivity can happen.
I wasn't setting you up, don't worry.
Yeah, no worries. But yes, currently I have looked at both the top 10% of data points, the top 15% and 25%. Recent work also in our lab has demonstrated that connectivity happens at all levels and different areas of connection.
Yeah, I was only mentioning that to connect to Sergey. So you're not correlating with the entire time course per se. You're picking out peak activity from that time course and then averaging or some combining time points together from that.
Yes.
So and that also those also give you that you're also computing the phase relative to those as well. So Sergey, that makes it's a little bit different than just a standard seed-based.
Yeah. Exactly. I was trying to understand what's the difference and how you get that. I think I got the answer. There is another question, if I may. The patterns, the temporal patterns. It's the slide after, I think, when you have multiple. Can you? And again, this I'm trying to understand. Can you use something like shapelets? Is that what you're after? You're trying to extract typical behavior, like temporal dynamic patterns, or I'm misinterpreting what I'm seeing?
Yeah, here is just the demonstration of global signal regression and no global signal regression on the data when QPP pattern analysis is performed. Here we can see that QPP1 without global signal regression is very similar to the global signal itself. What is being shown is that when regressed out, the first component of this windowed approach for QPPs is very similar to the second component without the use of global signal regression. What I'm demonstrating more thoroughly here is that this is also used in a secondary analysis by a complex PCA approach, which is data-driven rather than a windowed approach. And so this is just emphasizing the fact that the global signal is a largely coherent and well-established full brain signal that warrants further exploration.
Okay, I think I don't have questions right now. Maybe I can ask something later if I will be allowed.
Sure.
Yeah. Okay.
We won't cut you off. Maybe I will go next. So I have a few questions, just kind of get your thoughts on very interesting project, I think. One was sort of a simple one, just for my understanding. When you were showing the time courses for the control group and the AD group, I guess how are you controlling for the fact that the phase varies from subject to subject? Are you somehow because how do you ensure that the temporal averaging won't cancel, right?
Yes, that's a great question, and especially with this method, it has been shown that typically a window of 24 seconds is used in many of the analysis. However, this has been done throughout many different studies ranging from 20-second templates to 30-second templates. This is part of the analysis that we can be doing for this. However, a different approach that might help sort of be corollary to the results.
How do you define the template and how do you define the onset?
Yeah, so the template, so the template is hand-tuned. Specifically, we input that initially. However, we are able to look at many different windows within the 20 to 30-second time frame. But there are other data approaches or data-driven approaches.
I'm still missing something. So the time course you showed was just a window, right? It was just a 24-second window. But do you get one of those for each subject and then somehow combine them? Or are you computing are you somehow doing this separately for the two groups?
Yeah, okay, I get the question now. Currently, we are only using one template, and this is hand-tuned based off of a line of research that has previously been shown across many different data sets, especially in human data.
And a template is a spatial map, or it's a sequence of spatially evolving maps. What is a template in your?
Yeah, so the template is going to be a window of 24 seconds of the full brain volume and used as a sliding window throughout the whole data.
So it's a voxel-wise time series, basically, for that window, for the window.
Correct.
Yeah. Okay. Got it. So that's that template is defined from the entire data set, or it's defined from some other data set that you?
It is defined from many other data sets, but used for every data set, we try to use one that would correspond to the best anti-correlated signal between the default mode network and the task positive network.
Okay. Great. And then, so I think that answers my question. So you're using the same reference for everybody. So then you're just looking to get them relative to that change at each start and end within subject. So, yeah. Do you, for the one sort of minor terminology thing, which is you use the term biomarker and you sort of said it was a biomarker, right? And so I wanted you to maybe think about how do you define what constitutes calling something a biomarker, and because there's a lot of definitions out there, so you might want to clarify either we're looking for a biomarker and this is what will qualify it as such, or we're calling it that because of this, that kind of thing, right?
Of course.
I would suggest. Then your comparisons, I think you were focusing on the time series for the windows at the different bands. Can you also do can you pull out information, or have you considered pulling out information from the spatial aspects of this, and could you use that in this project as well?
Yes, absolutely. That is one of the pieces that I've not shown here due to brevity of the presentation. Looking at the spatial orientation of many of these signals in the overlap of potential disparities between the signals is of high importance. Specifically, we're able to remap this to typical standard space with the Montreal Neuroscience template, but that is not shown here during this proposal, but definitely a key aspect of the work.
But like I was thinking in particular, like the spatially evolving, the temporally evolving voxel-wise information. Could you summarize that in some way to get more of the spatial differences in between groups, or related to progression?
So So
I'm not trying to add too much. I'm just suggesting that because it seems like this is there's a lot of information you could use here, and I think there's a lot more you could potentially take advantage of. So
yeah, absolutely, especially
or maybe you already are.
Yeah, no, especially when thinking about how different information of the default mode network might not be consistent throughout the whole scan. knowing the way that it evolves over time spatially is also interesting. I don't have those results displayed here, but yes, that is a part of this research.
Yeah. I mean, because you're pulling it out, it's going to look a little bit like the template because you're defining it via that template, but you could look at individual subject representations and then summarize those in various ways. So
correct, like sort of like displayed here, where you see the spatial distribution of the default mode network and the task positive network throughout different brain structures and varying views.
Yes, but again, over time, right, as it evolves, right?
Yes.
Yeah, exactly. Oh, that is on the right. Yes, exactly.
And this is over time as well. So.
Yep. I see. Okay, and then one more question, which is the frequency range, the frequencies that you're looking at. So it looks like you're using the terminology that came from the ALF fields, right, where they kind of Zinnian's Woes stuff, which was done on slow TR data, right, 0.2 second data. And so have you considered? I know you have a couple additional ones you're looking at there, the full range and other things, but could you maybe explore that space a little bit? Because your data goes, you can go up to something like 0.7 hertz with your data if you wanted to, and you could also look at different, splitting it in different ways. So that might maybe looking at it just through the slow 1 through 5 kind of bands might miss something, right? Is what I'm saying. So there might be some other ways to filter it that might yield more information. So
yeah, that's a great point. I'd love to chat more with you about that, especially after the call or different things. But at least the choice for these specific bands were neurologically relevant factors that were able to be shown in previous studies, and just to at least be able to show the differences and reproduce their results or show insights to these more delineated signal bands might prove something interesting. So,
yes, and last question, you're for the work that you're doing, comparing the groups, looking at progression. Are you gonna be summarizing this using some sort of statistics, or are you going to do some sort of individualized prediction, or what is your kind of versus looking at the meat, sort of at the different, just plotting two plots, side by side doesn't necessarily tell you whether it's meaningfully different, right? So how will you be comparing these? Yeah.
So I'll be
sorry
specifically.
If I missed it, but
no worries. I specifically will looking at differences using t-tests, looking at the amplitude of the QPP patterns, the power of the signal variability, dwell times for each of the QPPs has shown to be an interesting comparison. We have several different histogram tests that we can do to look at the correlation, and then looking at the projected template between the groups is also a way to look at the differences between.
Okay, great. Okay, Shella or Jeff? Maybe Jeff. Do you want to go next?
Yeah, I have a few questions.
I mean, you know a lot about it, of course, but you know where all the bodies are buried. So why don't you ask him about one of those? No.
I think Shella knows where the bodies are buried.
Okay.
Well, I have a couple of questions. TJ, I was thinking the other day, of all the PhD students I've known, you are one of the most multifaceted. I mean, in addition to this great research that you have here, you have spent, as far as your entrepreneurial efforts, I know you spent a lot of time in that, advising teams and entrepreneurial efforts. And then complete the trifecta, you're like a super teacher, right? And which I think is accredited by all the great awards that you have been awarded by Georgia Tech and by ECE. And so this kind of motivates my two questions to you. Okay. So the first question, maybe I'll start entrepreneurial. So if you and Dr. Kelholtz were to commercialize this research, I guess my question to you is maybe threefold. One, who would be your early adopters? Two, what kind of friction or barriers do you foresee in that? And then maybe three, a little bit, which is maybe incorporated into that, is what would be your unique value proposition?
Yeah, no, those are great questions. The way that I would first start to find early adopters, of course, would be clinicians who would be on the side of collecting fMRI data, especially from those who have potentiation for Alzheimer's disease. The use of QPPs, or the spatially evolving patterns, are the ability to potentially show these early detection signs. And so by using this specific signal, we would be able to use, or clinicians would be able to use this to potentially give a patient early diagnosis.
So for those clinicians, can I interrupt you for just a second? So for those clinicians, I guess, so these would be people that they already maybe have a genetic predisposition for Alzheimer's, that they're trying to monitor a progression.
So
and that would really be that would be your first audience, I guess, in that regard.
Correct.
Okay.
In the clinic, when being diagnosed with Alzheimer's, they have to take in education, age, gender, and a variety of other factors that are combined into one score. And that score is then taken year over year to show either an increase or decrease in cognitive ability and show progression over time. A lot of times when AD is being diagnosed, they will not find clinically relevant data to support a diagnosis until the later stages or past MCI. So being able to find potential in healthy controls of patients over the age of 55 to 65 would probably be the initial beachhead market I would target. To areas of entry that might be difficult would be to make sure that the code is widely accessible and available to use. The code definitely needs to be able to have an ease of use. Potentially, if I was trying to market this, I would make GUIs. I would make it as easy as possible for users to be able to just click and play, rather than having to do much of the fundamental research that we typically do, where it's very hands-on for the code. But that would be probably our initial barrier to entry. Our market value is that this method is specifically robust enough throughout many different modalities, many different imaging practices, many different machines that are available on the market right now. One major advantage is by using the global signal as a potential marker for these types of progression delineations, is that signal is very easily extractable and can be used pretty readily, rather than using tens to hundreds of different regions of interest used to try to find different dynamics. Not saying that those are unwarranted, but potentially this signal can be a lower hanging fruit for people to use.
So you might have said this. So would this group, would they be getting enough functional MRI scans to be useful for you to track this, or would they have to up the number of fMRIs that they would have to get to use your method?
Yeah,
maybe I don't know. Maybe they already get a large number if they're in this group and it's a progression.
Yeah. So the reason why I think this is interesting is that over the last 10 to 15 years, ADNI has been collecting longitudinal data, and many times during a patient's study window over the 5 to 10 years that they've actually been participating, they have changed in diagnosis. One of my goals is to characterize the pre-diagnostic and post-diagnostic inflection point. And so by at least understanding where the patients might sort of encounter or be grouped up through based off of characteristics of the signal, it'll be a better indication on what they might need to follow up and test further for. And specific medications and treatments really do target mild cognitive impairment patients. So if we do find someone who is potentially on the cusp of a cognitive score, they can get an MRI. It might give indication that they are early MCI, late MCI, or even on that flexion point. that'll help at least distinguish between potential treatments they can get.
Definitely for this data, right? It's longitudinal data. So I assume, if they're part of the study, then they are getting more functional MRIs.
Mm-hmm.
Would if you were to commercialize this and you had clients, would they have, maybe you kind of said this, but would they have to get the same number that they would have this longitudinal study, or would it just be a sparser sampling for an individual over time to be enough for you to maybe have some information about, are they progressing, or how they're progressing?
No, that's a great question. And it's typically you want to meet a potential customer, potential user of this at their where they're at, rather than trying to change their behavior. Typically when you reach the age of 55, 65 and have a propensity in your family for Alzheimer's, you are typically seeing a doctor once a year, multiple times a year. During that, they can suggest getting MRIs if the need is there. But if I had to commercialize this, I would do a better investigation to see the frequency of a typical patient of 55 and older. So I don't quite have the frequency or the relevance to patients needing this, but the high advantage of using, or the great advantage of using fMRI and resting state fMRI is one, they don't need to be doing a task to understand the differences between the brain regions. They can sit still, where relatively that might be difficult for patients under neurodegenerative diseases. And two, it's relatively less expensive than many other distinguishing modalities within the field. So compared to PET, MRI is very cheap comparatively.
Okay.
And I believe covered by many insurances.
Okay. Interesting. Definitely interesting insight. So now you're going to take off your entrepreneurial hat, and I want you to put on your teacher hat, your teaching professor hat.
Mm-hmm.
And I guess I have to, you're so multidisciplinary. So I guess I have to pick a department. So I'm going to pick the ECE department, since I'm an ECE. And I know you've TA'd for ECE. If you were to, let's say you were an ECE professor, and you wanted to incorporate some aspects of your research, because it's really interesting application, and you wanted to incorporate that into the curriculum. Now I'm gonna take away grad classes, and I'm gonna take away like 4,000 level classes, because you say, well, I'm just gonna do a class that's very specific to this.
Yes.
Are there 2,000 and 3,000 level classes that you think that you could create labs for, or that to really show, to have this great application? And I guess that's my question. Like, how might you incorporate that to excite young undergraduates in this field in ECE? How might it incorporate into some of the classes?
Yeah, that's a great question, especially when thinking about sort of applications in digital signal processing, or DSP, which is typically where I think I fall into theory within ECE. Typically, these pattern, or the way, due to the complexity of brain data and the nature of MRI itself, typical analysis does suggest static functional connectivity, meaning they take the entire scan and use it to find the functional connectivity of the brain. Much like the initial example I gave at the beginning, where the sliding window correlation is showing a windowed approach for the functional connectivity. This is not taking into account the temporal or the time dynamics within the scan itself as these patterns evolve. If I had to put this into sort of a class, I would probably look either at time-varying analysis in terms of a DSP unit, or some kind of I guess I haven't looked at the undergrad catalog, but if there was a class typically dedicated towards DSP or applications of DSP, this might fit into a great unit of both looking at time evolving methods, spatially evolving methods, and then specifically combining them together, as there is a couple of different approaches that you can use that are just outside of this scope, but are still prevalent within the field.
I guess, and I know I'm going to pass the baton here because I don't want to take too much time, but I guess the question, it seems to me the question, which is a lot of times you have this, right? If you want to incorporate this into like early DSP class, or even like a programming class, or with different labs, there's a lot of complexity, right? There's complexity to these signals, these bold signals, and the data that you have. I guess the real question is, well, is there some way that you could simplify it, for them to do? I mean, you're looking at bandpass filters, right? Or you're looking at different filtering. Is there some interesting opportunity there that they can do some computation, and you can say, hey, look, this is,
Yeah, I would probably
showing.
No, just in terms of like always having a limited window of being able to teach various students, and you have to take into consideration the time it takes to process and analyze the data itself. I would give them canonical networks that were smaller and well established, much like YAO, where there's only 7 networks to be analyzed. That way, the complexity of the data can at least be interpreted by the students. But yeah, typically with this type of project, yeah, I know for my machine learning and for my DSP courses in grad school, I did use fMRI data. So I would say they would probably be more final project based, or like a capstone of the class project, just to be able to look at this data due to its complexity. Simulated data is great as well if we're trying to find specific dynamics between certain networks, and those can be generated with a variety of different methods.
Okay, fair enough. I know that I'm kind of putting you on the spot there a little bit, but I'm just kind of.
No, it's a great question.
Because I know you're such a great teacher. So anyway, so those are my 2. I'll pass the baton.
Okay, and so Shella, last to you. Just a time check. We've only got about 11 minutes left, so but I figured you have probably already asked all of your questions, but you may have a few that you want to highlight here. So.
I pretty much have, but the one thing that I want to ask is, I think this comparison between frequencies, atlases, TRs, this is all important, and it's nice to have a systematic look at these things. But I think something that didn't come across in your presentation is, how are you going to decide what's the best parameter? Or even beyond that, how are you going to decide what's good enough? If somebody asks and says, can I do this on data with a TR of 3.5 seconds? How do we know?
Yeah. So typically when we're looking at many of these parameter designs, we're looking at the anti-correlation between the default mode and the task positive. And typically we do that in healthy patients and healthy subjects to find this intrinsic brain network interaction. As we go through and do do come up with the results from the larger data sets, it will at least show whether or not these higher TRs, for example, where there's a deprecated window length to find of the template to find these anti-correlated signals, it might not be enough. However, I do think this is a great question and probably needs to be more explored as we continue this analysis. But understanding better recommendations will be an aim towards the publication of these results.
Okay, well, I can stop with that in case anyone wants to revisit other things, because you and I, of course, have discussed it a lot.
Yeah.
Great. So in the interest of time, why don't